Good morning, students. Today we will discuss about the cells of acquired immune system. Cells of acquired or specific immune system. Cells of acquired or specific immune system. In this, two major groups of cells. Two major groups of cells are involved in acquired immune system of the body. Two major groups of cells are involved in acquired immune system of the body, which are these two major groups, which are: first is the lymphocytes or lymphocyte cells, and second is the antigen presenting cells. The cells of acquired immune system. In this acquired or specific immune system, two major groups of cells are involved in acquired immune system of the body, which are: first is the lymphocytes and second is the antigen presenting cells. Then we will see first group of cells, that is the lymphocyte cells. Lymphocyte cells. These are the chief cells of. These are the chief cells of acquired immune system. These are the chief cells of acquired immune system of the body. They are of two types. They are of two types. They are of two types. First type is the T lymphocytes. First type is the T lymphocytes, or which is also known as the T cells. which is also known as the T cells. Then second type is the B lymphocytes. B lymphocytes or B cells. Lymphocyte cells are the chief cells of acquired immune system of the body. They are of two types. First type is the T lymphocytes or T cells, and second is the B lymphocytes or B cells. Then both this type of cells are produced. Both the types of cells means the T lymphocyte cells and B lymphocyte cells. Both the types of cells are produced from stem cells. Are produced from stem cells in bone marrow by a process of hematopoiesis. Both the type of cells are produced from the stem cells in the bone marrow by a process of hematopoiesis. Some of them are undergoes differentiation. Some of them undergoes differentiation in gut associated bursal lymphoid tissues or B cells. Both the types of cells produced from the stem cells in bone marrow by a process of hematopoiesis. Some of them undergoes differentiation in gut associated bursal lymphoid tissue or B cells, or which is also known as the B lymphocyte cells. B lymphocyte cells. Other of them differentiate in thymus gland, hence known as T lymphocytes. Or T cells. Some of them undergoes a differentiation in gut associated bursal lymphoid tissue, which is called as the B cells, and other of them differentiate into the thymus gland, hence known as T lymphocyte or T cells. Then the mature lymphocyte cells pass into the mature lymphocyte pass into the body fluid, that is the blood and lymph. The mature lymphocytes pass into the body fluids, that is the blood and lymph. Then many of them stay in the lymphoid nodes. Many of them stay in the lymphoid nodes. Then the B lymphocyte cells and T lymphocyte cells form. Then both the cells, that is the B lymphocyte cells and T lymphocyte cells, both the cells are formed. Humoral or antibody mediated immune system, both the cells, that is the T lymphocyte cells and B lymphocyte cells, are formed. Humoral or antibody mediated immune system and cell mediated immune system respectively. Then both the immune systems needs the antigens. Both the immune system needs the antibody mediated immune system, and the cell mediated immune system needs the antigens to come in the action, but they respond in different ways. Then next is the mechanism of T lymphocyte to antigen. Next is the mechanism of action of T lymphocytes to antigens. Mechanism of action of T lymphocytes. On coming in contact with an antigen, a T lymphocyte form a clones of T cells when the T lymphocyte cells which are coming in contact with antigens. These T lymphocyte cells form a clones of T cells. Forms a clones of T cells. The clones of T cells which are similar. The clones of T cells which are similar but they perform a different functions. These clones of T cells are similar. They are similar but perform different functions. The clones of T cells which are similar but perform different functions. The clones has 4 types of T lymphocytes. The clones has 4 types of T lymphocytes. Clones has 4 types of T lymphocytes. First lymphocyte is the helper or T cells. Helper T cells or helper T cells. These helper T cells are produced lymphokines for performing lymphokines for performing several types of functions. These helper T cells produce lymphokines for the performing different or several types of stimulation of cells, then macrophages, etc. The first type of closed or T lymphocyte is the helper T cells. These cells produce lymphokines for performing several types of functions or different types of functions like proliferation of other T cells, stimulation of B lymphocytes, stimulation of B lymphocytes or stimulation of B cells, then macrophages, etc. Then second is the killer cells or cytotoxic cells. Second is the killer cells or cytotoxic T cells. Killer T cells or cytotoxic T cells. Second is the T cells or cytotoxic T cells. These killer T cells or cytotoxic T cells which directly attack and destroy invading microbes. They are directly attack. They are directly attack and destroy invading pathogens. Invading pathogens which are infected body cells. Invading pathogens which are infected body cells as well as a cancer cells which are infected body cells and cancer cells. Then third is the suppressor T cells. Third is the suppressor. Suppressor T cells. These suppressor T lymphocyte cells which suppress the immune system, against attack on the body cells, Then fourth is the memory T cells. Then fourth is the memory T cells. These memory T cells or these memory T lymphocyte cells are previously sensitized cells. These are the previously sensitized cells which retain sensitized memory, which retains the sensitized memory. Then next is the mechanism of action of B lymphocyte. Then next point is the mechanism of action of B lymphocytes. Mechanism of action of B lymphocytes. B lymphocytes are sensitized directly by both these B cells, which is sensitized directly by both antigens as well as helper T cells. The next point is the mechanism of action of B lymphocytes. In this, the B lymphocyte cells, which is sensitized directly by both antigens as well as helper T cells. The activated B lymphocyte cells multiplies. Activated B lymphocyte cells which are rapidly or fastly multiplied. Which is rapidly or fastly multiplies to produce a clones of, to produce plasma cells and memory B cells and memory B cells. In this mechanism of action of B lymphocytes, these B lymphocyte cells are sensitized directly by both antigens and helper T cells. These activated B lymphocyte cells, which are rapidly or fastly multiplied to produce a clones of plasma cells and memory B cells, then which are circulated through a body fluids. The plasma cells produced a specialized glycoprotein molecules. Specialized glycoproteins molecules known as antibodies known as antibodies. These plasma cells produce a specialized glycoprotein molecules which is known as the antibodies, and these specialized molecules which are circulated into a body fluids like a blood and lymph. Then the antibody molecules may be bind with a cell membrane. Antibody molecules bind with a cell membrane. Or may be free. These antibody molecules bind with cell membrane and may be free. The free antibody molecules have three main functions. The free antibody molecules have three main functions. Molecules have three main functions. These are first function of free antibody molecule is the agglutination. Second is the opsonization, and third is the neutralization. First is the agglutination, second is the opsonization or coating of bacteria, and third is the neutralization. Opsonization or coating of bacteria, and third is the neutralization. Third is the neutralization. The first function of agglutination of particulate matter including bacteria and viruses. Particulate matter including bacteria and viruses. These mass the particulate matter including bacteria and viruses. The immobilized mass is then anchored by phagocytes. These immobilized mass then anchored by phagocytes. Then second function is the opsonization or coating of bacteria to facilitate their subsequent phagocytosis by macrophages. Opsonization or coating of bacteria to facilitate their subsequent phagocytosis by macrophages. Then the third function is the neutralization. The toxins released by bacteria. That is the tetanus toxin. These are the three main functions of pre-antibody molecules. First function is the agglutination. Second function is the opsonization. Third function is the neutralization. Each antibody is a specific for a particular antigen. Each antibody is a specific for a particular antigen. Each antibody is a specific for a particular antigen. Then the second group of cells of acquired immunity system. The major second group of acquired immune system is antigen presenting cells. Antigen presenting cells. Firstly, we study the first major group, that is the first lymphocytes cells of group, and then second, the major group of acquired immune system is the antigen presenting cells. Antigen presenting cells which engulf invading pathogens, which engulf invading pathogen by a process, the antigens by the process the antigen presenting cells which engulfs invading pathogen and process the antigens. Then the processed antigens are present on their own surface. And these cells able to deliver, and these cells are able to deliver a stimulating signals, These cells are able to deliver a stimulating signals for activation of helper T cells. These antigen presenting cells are able to deliver a stimulating signals for activation of helper T cells. These are the two major groups which are involved in the acquired immune system. Thank you.